So I'm very pleased to introduce Adele Malone from Arizona State University, just north of us down here in Tucson. And Adele's currently a master's student in Dr. Krein Pyman's lab. Did I do it? I got it right?
Yeah.
Um, and at the same time, because that's just not enough, she's also working as a lab technician at the Maricopa County Environmental Services Vector Control Division. Um, before she saw the light and started working on mosquitoes, apparently you like bees are kind of nice. Was working on native bees in North Dakota State, where she got her bachelor's degree. And now we're going to learn about the work that you've been doing, looking at quantifying time, location patterns of human mosquito interactions to inform operational control activities and public education programs. Thanks so much, Adele.
Thank you very much for the introduction. I'm going to share my screen real quick. All right. Yes. As Kathleen said, I am Adele Malone. I am working in Dr. Krein Pyman's lab at ASU, and this is a vast majority of what I do. Um, I will say that everything I am presenting here today is through ASU. It is for my research, and it is not representative of what goes on at Maricopa County. Um, but my project is quantifying time location patterns of human mosquito interactions to inform operation control activities and public education programs. So let's get into it. So those of us who already are in Maricopa County, we know it's massive, but it has grown considerably even within just the past 10 years. So on the right, I have a figure that goes all the way from 1912 up to the 2010 census year. So that is dark purple all the way up to dark brown. And it's detailing the growth patterns of the Phoenix metropolitan area in Maricopa County. And so you can really see the massive expanse of the area, and you can also see exactly where it's going to grow into in order to fill all of those gaps. And so this growth, you can physically see how it's been spreading, but you can also look at permanent residents. So in the 2010 census data, we have around 3 million people. And then in the 2020 census data, we have 4 million people. And this is just counting permanent residents. So it has nothing to do with people who are here on business or doing work. It doesn't represent all of the students that are in Maricopa County. So they're here for like 8 months out of the year, majority of the year, but they're not considered full-time residents. It doesn't count snowbirds, people with double residence. And then it also is not accounting for the growth that's happened even just within the past 3 years, which there is a lot of growth. And so with that growth comes a lot of opportunities for mosquito-human interactions. The way that urban landscapes are set up is pretty prime for mosquito breeding. I have a couple of examples. So we have urban infrastructure, so our roads and our sidewalks that have potholes that have water in them. There's also our sewer systems, our water systems. Those are really good for breeding. A lot of human recreational activities are also really good for creating human mosquito interactions. I have an image of a man golfing, which Phoenix metropolitan area also has a ton of golf courses. There's also backyard landscapes, backyard decor, natural spaces, or even just parks in the city, areas in particular with natural bodies of water. And then almost most notably, we have a pile of garbage, which actually presents a ton of breeding opportunities. It's one of the fun facts of like you can take a bottle cap from a water bottle, have it upside down, put a little bit of water in it, and an Aedes aegypti will find it and breed in it and create a massive problem for you. So these urban activities, these human behaviors present a huge problem. Um, and ultimately, these mosquito human interaction possibilities are a public health concern. And so you can see on the top half, I have West Nile virus information. In Maricopa County in 2021, we throughout all of our testing throughout that entire year, had 483 positive pools. And so that is possibly more than just 483 positive mosquitoes, because up to 50 mosquitoes can be put into a tube. And so that's pretty high, considering in 2022 we only had 47 positive pools. But in addition to just what has happened at the Maricopa County Vector Control, you can also look at human cases where we had almost 1,500 human cases. 64% of those were a neuroinvasive type rather than non-neuroinvasive. And this is well above the national average. It's actually supposed to be less than 1%. So we have a particular endemic strain that is very threatening. In addition to West Nile virus, we've also had St. Louis encephalitis, and most recently, we have had our first evidence of locally acquired dengue. And so, considering the metropolitan area is in a like oasis zone and is expanding, creating more opportunities for mosquito breeding, this is of huge public health concern. And so our governmental pest control services and education services work to inform the public and fight mosquito breeding and feeding opportunities. And so this is primarily through IPM, or integrated pest management, which is like a multi-level method of addressing pests. Ideally, it's with the most ecological and safe methods before you do any sort of chemical method. And so really this includes like drain monitoring, biocontrol, public education. You do a lot of citizen outreach. And then eventually you do go towards like adulticide fogging or space spraying. But for these methods to be effective, our governmental pest control services and educational services need to be properly informed so that they can continue to use the safest and most effective methods. And so one of the primary methods of monitoring these mosquitoes and the disease abundance is through routine trap monitoring. So this is an image of over 850 traps that Maricopa County sets every single week. And each one of these traps encompasses roughly a square mile. It varies depending on where the metropolitan area has spread out to. But the thing is about each of these traps, they can be placed in like a yard, someone's front yard. It can be placed at like a public facility, such as like a fire station. But in each of these square miles, there's a lot more than just houses, or there's more than just fire stations, right? So there's going to be like parks or pools or businesses or drainage ditches or natural areas or apartment buildings. So although they're very good at getting a general representation of where mosquitoes are, they're not really painting more of the picture for the nuances of human activity or where humans are at certain times of the day, which is why it is important to be able to look at mosquito activity. And so considering when mosquitoes are active, it is important for a few reasons. So vector control services need to know the prime times for treating areas with mosquito abundance and disease presence. The effectiveness of treatments is largely dependent on whether spray can come into contact with the bodies of mosquitoes. So sprays need to be done at the same time mosquitoes are active. Then in addition to treatment, prevention on the human side is also very important. So citizens need to be informed about mosquito risk variation throughout the day so that public and private events can be appropriately held. The timing of these events can vary depending on where they're being held. Citizens can then use informed risk and better respond to risk throughout the day. And so ultimately, this also means that at-risk groups can be identified and protected. So many citizens are at risk of mosquito-borne vector diseases, and identifying mosquito activity time or mosquito bite time, prevention strategies can be targeted to assist these groups during those peak mosquito activity hours, thus preventing lots of illness. So in order to study when mosquitoes are active, I'm also focusing specifically on, is mosquito activity time different between species and sex, and does activity time change throughout the year? And so in order to start addressing this, I took data over the past 4 years for all 850 plus of the traps that Maricopa County sets routinely. And I was able to identify the traps that are consistently striking positive for West Nile or SLE, dengue, chick, Zika. All of the herboviruses that Maricopa County routinely checks for. And then along that gradient, I was able to establish 5 different locations to be able to monitor for deal activity. Okay. So from left to right, you have 1, 2, 3, 4, 5. And there's a lot of text on this page, I know, but it's really just a description of each of the locations that you can kind of get a clearer picture of what the data eventually is going to mean. So for example, the first one really is a series of rented duplexes, not a lot of green space. The second one is a residential area, lots of individual homes. The third one is also a series of rented duplexes, much more shaded near a pool. The fourth location is a residential area right next to a park, and it also has a pool. And then the fifth location is a residential area with a lot of shading and a lot of greenery in the yards. So there's a pretty good spread of types of living spaces and areas near the traps to give us a pretty good clue of deal activity time. So at each of these locations, there is a rotator trap being set up. And so it is a 8-hole contraption that can rotate to pre-programmed times. It's set up on a tripod to be at the appropriate height. Then it has an EVS trap at the top. So it is primarily designed for capturing Culex mosquitoes. It will rotate for us on the hour. And although there's only 8, we did still do once every hour. It's just that there would be switch outs every 8 traps to be able to complete that full 24-hour sequence. And so we would set up our trap underneath a bucket of dry ice for the CO2 to attract the mosquitoes. And it would be preferentially in an area where it could be shaded for a large majority of the day. And then we would set it up at 6 a.m., do a 2 p.m. exchange, a 10 p.m. exchange, and then the next day at 6 a.m., we would then remove the trap to capture that full 24-hour time. We also did record local weather so that we could hopefully identify reasons why one location would be, I guess, very proactive, able to capture a lot of mosquitoes, and some locations might be suffering a little more, looking for just differences between areas based on weather. And so we were considering, what do we think we would expect to see? So this is on the left, that is, there is a graph from a paper that was released this year. It was a study done in 2022 looking at deal activity patterns of mosquitoes in Florida and Texas. Although it did focus primarily on Aedes aegypti, so a little bit the opposite type of trap as I'm using. So it used BG Sentinel traps, and they did theirs over 3 days. I think it was once a month. And what they ended up finding is that Aedes aegypti were diurnal. They would have 2 peaks during the day, one very early and then one kind of later, but still just in the evening. And then looking at Culex mosquitoes, it would be kind of like an overnight thing. They would be more nocturnal and more consistently active, just not in the middle of the day, not during peak sunlight hours. And so we would kind of expect to see something very similar, but it is important to keep in mind that these areas are both more humid and receive more rain than Arizona does. Okay. And then because Arizona has a lot of Culex mosquitoes, we also thought about what Culex mosquitoes would be influenced by. And Culex mosquitoes are ornithophilic, meaning that they preferentially feed on birds rather than mammals, and specifically rather than humans. And so we would think that Culex mosquitoes would be more active at hours where birds are more prone. And so more birds are prone at night because they're sleeping. And so Culex mosquitoes would likely be active towards the evening hours or at night while birds are asleep. And then another thing to consider would be seasonal differences or differences throughout the year. And so this was a study that was actually done in collaboration with Maricopa County, looking at the 2011 to 2021 mosquito abundance rates so that they could look at monthly abundance of the 3 species that Maricopa County receives the most of throughout all of their routine traps. So they're looking at Aedes aegypti, Culex quincunfasciatus, and Culex tarsalis. And so they did also compare it to temperature and rainfall, but really what we're looking at here is that there are 2 peaks for mosquito abundance. You have one in the springtime and then one in the fall. But you're really not seeing much happening during that kind of like the peak of the heat. So that June, July, August, leading into August dip, that's when it gets really, really hot. And then you're also seeing low counts during the winter time. So going from November to December, January, February, it's also going to be really low. And so we would think that mosquito counts will be lower in the middle of summer and the middle of winter, which also can impact mosquito behavior. If you have a mosquito that prefers warmer temperatures, maybe they'll be more active during peak daylight hours during the summer, or sorry, peak daylight hours during the winter, whereas during the summer, peak daylight hours might be too intense, so they'll be active either slightly before or slightly after. Okay. And so before we really dive deep into the results that I'm presenting, I wanted to go over this graph. It's not super intuitive. I have 6 a.m. at the very top, and that is because 6 a.m. is when we started our trap times. And so it's going to go from the initial 6 a.m. all the way back over to the collection 6 a.m. So that's going to be the deal activity. I do have a sun and a moon located on the right and left sides, respectively, to represent that for the most part, the 6 a.m. to 6 p.m., that's going to be the sun up time, and the 6 p.m. to 6 a.m. on the left side, that's going to be the sun down time. Mostly just to help figure out when we would have daylight. And then when you're looking at these numbers don't mean anything, it's just an example to help read the graph. But when we're looking at peaks for mosquito behavior, these numbers represent how many were caught at a specific time. So if we're looking at this means that at 8 a.m., 4 mosquitoes would have been caught. And here, if we're looking at 4 p.m., that means 6 mosquitoes would have been caught. And then, for example, 5 a.m. and 6 a.m. both would have received 2 mosquitoes each. Okay, so to give a little bit more clarity as to why we're seeing what we're seeing, this is first looking at our collection throughout the entirety of the mosquito season. So we started in April and we're ending actually this week. This is the last week of November. So this is our last collection week. So weeks 30 to 35 are incomplete or only partially complete just due to, finals are coming up. I have to split my time, so not all the data is logged, but this is very accurate up until week 30. But you can kind of see that some locations are more prosperous than others, but really what we're seeing is where our peaks are for mosquito abundance. And so this is an Aedes aegypti. And for the most part, you're really seeing peaks just before week 15. And it's shown here, this mimics the Maricopa County collaboration study where you're seeing a peak before and after the height of the heat for Phoenix. And then we are looking at those deal activity graphs here. And each location is a little bit different. But looking at 6 a.m. at the very top, you're able to see that for the most part, locations are still receiving a peak pretty early on in the morning, a lot near 5 a.m., which during the summertime, Phoenicians know that you need to get out before it really hits like 7 a.m. if you're going to do anything extensive during the day. So it does get hot quite fast. And if we combine everything, there is still a very strong peak at the 5 to 6 a.m. There were not a lot of males throughout the entirety of the experiment, and that is kind of just dependent on where your traps are going to be placed. Aedes aegypti mosquitoes in particular are not really the most adventurous flyers. They don't really go like, I think it's a mile from where their breeding site is. So unless these traps were located very close to their water source, the breeding area, they're not going to capture a lot of males. So it's not surprising we're not seeing that. All right, so then moving on to Culex tarsalis. You can also see some peaks here pretty consistently across the board. We had the most number of females caught around week 5. Really, there's not a ton of tarsalis being captured in a lot of these areas. Yeah, only 41 total. The biggest peak is still over near 5, and we're not seeing another huge peak, but these mosquitoes are very dependent on kind of like the murkier swampish water, which we got hit with a pretty big drought. So we also are likely to not see a ton of these. But when we look at their deal activity, this is what it ends up looking like. Again, very low counts for locations 1, 2, and 3, but locations 4 and 5, you can see some stronger peaks, more consistent catches at very specific times. And then looking at everything together, it is during the nighttime, either super late or super early, depends on how you look at it. But we are seeing peaks at 11, 1 to 2, and then 4 a.m. And then we're also looking at I'm calling it a QP complex. Culex quinquefasciatus and Culex pipiens are very, very similar physiologically. So just looking at them, it's very difficult to be able to differentiate them, even using taxonomic keys. So we do plan on sending out some samples in order to get them genetically tested so that we can get a clear picture of what is happening in Maricopa County. Both quinks and pipiens are able to transmit West Nile virus, but there's kind of evidence showing that pipiens might be more effective at transmitting West Nile virus. But again, they're both ornithophilic, so their behaviors are likely to be very similar. And we by far caught the most of the QP complex out of any other species that we were looking at. Locations 1, 4, and 5 are all seeing peaks a little earlier on, but ultimately we're still seeing just one peak before it gets really hot, and it's kind of tapering down after that. But 169 total were captured. And then this is their deal activity. This is very similar to what we did see for Tarsalis, despite having so many more quinks. It is reinforcing kind of that very late, early morning patterning where you're seeing a peak at 11, 1 to 2, and then again at 4 a.m. Right. And then there were only 7 of them, but I thought I would mention it. There were some Saurophora found at location 5, which was the very shaded location and residential area. You know, there's only 7, so it's not something to really write home about. It's not that significant. There's not much we can say about it, but if we were to represent it, it would look like this. And it is interesting to me, though, that when we started really capturing the Saurophora, it was right after and during the time where we were hitting peak heat. And so the other species went down a little bit in their abundance, but Saurophora is kind of started picking up, you know, as much as only 7 samples can. So what does all of that mean? Okay, well, first and foremost, the results are not final. Data is still being collected. So those images are likely not what they'll end up looking like overall. But they're a pretty good clue of what we have so far. And it is it is looking like other studies for what Aedes behavior is like and what Culex behavior is like. But ultimately, there were a lot of environmental impacts that led to a very low collection season. It wasn't just us. Maricopa County Vector Control also had below average capture rates for mosquitoes, but ultimately it was very low rainfall and very hot weather. There was like a month over above 110 degrees Fahrenheit, most of which was near 120 degrees Fahrenheit. Which at that point, it's just super inhospitable for mosquitoes, both the adult stage and the pupa and larva stages. You know, the water just gets so hot that development is severely harmed. So there just was not a lot of proliferation happening at that time. But now what do we do? It was a crappy collection year. What do we do moving forward? So we're going to do it again. Locations are pending. We're going to hopefully look at our trap design to see if there can be fewer net switch out times. And then we would like to be able to do it again. And hopefully this next upcoming year will be more normal for the climate. But you know, maybe this year, 2023, will be reflective of what seasons are going to look like moving into the future. I mean, there's been some signs showing that the Pacific Southwest is going to get hotter and it's going to get drier. So it may just be the first year of many showing new patterns. But ultimately, after these 2 years are finished, we would like to combine the mosquito deal activity and be able to look at human deal activity and find the ideal transmission intervention. All right. So ultimately, mosquitoes can be found everywhere throughout everyone's lives. So it's important that mosquito deal activity be monitored. It's going to be very important for mosquito transmission intervention. It informs pest control and education services. It can help to identify and protect at-risk groups. And ultimately, citizens are then able to use informed risk assessments to go about their day. That is everything that I had to say. I want to give a thank you to the Pacific Southwest Center of Excellence in Vector-Borne Diseases for funding this work and giving me the opportunity to present. I really appreciate it. Also, thank you very much, Maricopa County. You have made this so possible. And like you've shared so much data to give me so much context. I really appreciate it. And then all of the project volunteers that ran each of these 5 locations, people who helped babysit traps, were primary carers for traps. I really, really appreciate everything you've done.
All right, thank you so much, Adele. I think we have 2 minutes for questions. What questions do people have for Adele? I know that there were some questions in the chat about I can't remember who it was, but whoever had those questions, could you ask them again?
I think that might have been me. Really great work, Adele. I was
Thank you.
wondering about the trap that you used. Is it biased towards females, especially considering that you're using dry ice as a lure?
Yes, absolutely. It is the females who are looking for blood donors to lay eggs. And so they're going to be more attracted to those bioattractants such as CO2 to show that there is like a blood donor in the area. In addition to how females are more likely to fly out of the regular breeding range to be able to look for hosts. So I would say it is biased towards female collection, but we do care more about, for the purposes of creating an intervention for arbovirus illness, we care a little bit more about being able to find female behaviors because that's how humans are getting ill.
Okay. If I may ask one more question, I'm curious about your Serophora collections. Although the numbers were low in your current collection or recently past collections, do you have any thoughts on how they might affect the population of your target species and how that might be incorporated into the program that you're developing or contributing to?
Can you repeat the very first half? I couldn't hear. I'm sorry.
The number of Serophora that you collected, assuming those are high, since you had we've had an unusually poor mosquito collection season, if these numbers are high enough to impact the population of your target mosquito species, how would that inform the program that you're developing or contributing to?
No, that is a very good question. I'm actually not entirely sure where they came from. Location 5, where the Serophora were found, didn't have freshwater or flood watering around it. So I'm not sure how the floodwater mosquitoes made it in there. So I would have to assume that they are in the pupa and larva stage out competing. At least during those temperatures, that really extreme heat, they were out competing the Culex mosquitoes. In terms of how it's going to impact the collection moving forward, just considering it was only at one location, I don't know how much we can read into it. Maricopa County does have traps set up at river bottom where there are a lot of Serophora and Culex mosquitoes, and they kind of just take turns having cycles throughout the year. So they're not really wiping the other species out at those locations. It seems more like they're finding more optimal breeding at different times of the year.
I think we have time for one more question from Neil.
Yeah, so nice talk, Adele.
Thanks.
We've been collecting mosquitoes out here in Salt Lake City on the wetlands, monitoring when they're coming into traps in with a BG counter and sentinel trap.
Mm-hmm.
So we get finer scale data than you were getting with your rotator trap.
Mm-hmm.
But this question was also in the chat as well, actually. But my question is, have you thought about sort of showing the data with respect to sunrise and sunset times, which are going to vary considerably throughout the seasons that you're collecting?
Yes, no, I have been recording it down. I've been recording it along with weather. I'm also able to go back and look at whether or not there was, say, a full moon. I did see I read a paper that looked at the possibility of full moons impacting Culex nighttime behavior. And so I will be looking into that, but I don't have any sort of imagery or I guess say anything to say pertaining to that at this moment.
Okay, thanks.
Yeah.
All right, I know there are more questions, but I really want to make sure that we give our second speaker his full 25 minutes. So I'm very pleased to introduce our other scientist from Arizona State University, Don Ward, who is currently doing his PhD on insecticide resistance and the evolution and ecology of field mosquito populations. Before coming here, he did his bachelor's at Auburn and his Master of Science in Public Health and Tropical Medicine at Tulane, and also worked for 4 years on Anopheles stevensii, which is now becoming the Anopheles vector of the malaria vector of the hour. And Don is going to talk about surveillance and operational research for targeted mitigation of insecticide resistance in Maricopa County.
Thank you for the introduction, Kathleen. Can everybody hear me?
Yes.
Great. Just as Kathleen said, my name is Don Ward. I'm a second-year PhD student at Arizona State University, working in Dr. Sylvie Hoban's lab. Been working with Aedes aegypti for a couple of years of my quote-unquote career, and Anopheles stevensii, and just started working with Culex mosquitoes. So a lot of my work that I'm going to be presenting today will be working with or talking about Culex quinquefasciatus and insecticide resistance, and another project going on there. So just an outline for my talk. Today I'll be talking about a little bit of the project that I've been starting. I started up last year involving insecticide resistance surveillance in Maricopa County. And then on my PacVec project, the operational field and semi-field studies involving insecticide contact efficacy. So going into my first project, there's a real issue, as I think everybody in the vector control, vector field knows, with insecticide resistance. Chemical interventions are probably the fastest and most efficient methods that governments use to control disease vector populations, in contrast to biological means and physical means, such as just container control and dumping over containers using gambusia fish, etc. And primarily, vector controls use pyrethroid insecticides, such as permethrin, deltamethrin, etc. But with that use of chemical intervention comes insecticide resistance. So as mosquitoes are dumped with insecticides, seemingly aimlessly just to control their populations, they contain different resistance mechanisms, either genetic or metabolic, that will allow some to survive and a lot to die out. The survivors will then populate the area in which or outcompete and populate the area in which the heavy spraying was performed, and then we'll have resistant populations in certain areas. Going a little bit more into the resistance mechanisms, the first project that I'm working on involves looking at both genetic and metabolic target or resistance mechanisms. Specifically, with the genetic mechanism, there's a gene in mosquitoes called the KDR gene, which there are 3 primary mutations, 2 of which are much more relevant than the other, the V1016I and F1534C mutations, which we use to screen for different levels of resistance in our lab, and also the V410L, which is, at least in the literature, not seemingly as important to resistance, but we still have begun to screen for it as well. And another resistance mechanism, which is primarily found in Culex mosquitoes, is our metabolic resistance mechanisms, particularly involving enzymatic resistance. And so you'll have different, which I'll discuss a little bit later, different enzymes that degrade pyrethroids at varying rates, which are, the enzymes are also naturally coded by genes, but I'm going to specifically talk about the enzyme activity itself. So the gap we're really trying to understand in this research is an incomplete understanding of the genotypic-phenotypic relation of genetic and metabolic resistance mechanisms with resistance phenotypes, particularly in primary disease vectors we have in the United States, Aedes aegypti and Culex species. In Maricopa County, we have Culex tarsalis and Culex quinquefasciatus, amongst others. But those are the 2 primary vectors of West Nile in the area. And I'd also just like to look more closely at the incomplete Culex resistance profiles in Maricopa County. I think a couple of studies have done some work, at least with the phenotypic resistance, but I'm particularly looking at both phenotypic resistance and how that relates to their metabolic resistance. Looking at this figure from one of our collaborators, Alden Estep at the USDA in Florida, you could see how he's guiding me a lot on the work that we're doing here. He mapped resistance profiles and genotypes across the state of Florida. They get collections from every county in Florida and then are able to genotype them. And you can see just how resistance, the darkest blue is the most resistant genotype. You can see how resistant a lot of these populations are. And he's also participating, and along with us, with another grant that we're working on with characterizing the same are doing the same kind of surveillance in Maricopa County. Which leads me to my questions. Not really going to focus on number 2 because, like I just said, there's another grant that is going on that's actually doing more of the surveillance. But I particularly want to know, how do insecticide resistance genotypes in the KDR gene and metabolic phenotypes influence survival in both Aedes aegypti and Culex quinquefasciatus mosquito populations, respectively, in Maricopa County? So the work I've been doing starting last year, where I was doing a lot of field collections of Aedes mosquitoes in the beginning, all of last fall, rearing them to adulthood and performing a dosing assay called a topical bioassay, which I'll go over, followed by genotyping the mosquitoes that are come out of those assays. I also did a little work on producing F1 mosquitoes to see if there was a difference between the field caught and the F1 mosquitoes. Don't really have too much data on that, but just wanted to mention it. And then following that up after the dose response assays with genotyping of the Aedes aegypti mosquitoes and enzyme activity assays. This year, I've been focusing a lot more on Culex. I've been doing field collections all of this fall in 8. Actually, originally had 9 field sites, but 8 heavily producing Culex field sites in Maricopa County in the Phoenix metropolitan area, mainly in this East Valley, what we call the East Valley area. It's pretty close to the Arizona State University campus, and is actually like a 3-hour round trip when I would go out and do these collections. But was able to still collect a good amount despite, like Adele was saying, we've had pretty dry weather here, but at least towards October and November, I've been actually able to collect a lot of mosquitoes in these BG GAT traps, which we modified not just to collect the adults with the little sticky paper, but we, as a advice from Jim Will at the Maricopa County vector control. We would just put these out with water and rabbit pellets, and they were perfectly able to collect a lot of Culex egg rafts, up to I think 60 at one point on one site, which was really shocking for me for this area. So despite the dry weather, we still have active mosquito breeding going on. The topical bioassays that I mentioned is a dose response assay where we use this repeat pipetter to apply approximately, I think, half a microliter of insecticide. I'm specifically using deltamethrin pyrethroid onto the abdomen of a mosquito. And then we wait about 24 hours to assess their mortality so that we can establish dose response. And I'm particularly doing it by site for both the Aedes and Culex mosquitoes. But particularly with the Aedes, since I'm able to do the KDR milk curve analysis assays, or qPCR analysis. I'm able to use both live and dead mosquitoes in that analysis. So the particular kind of data set that we're working on getting is characterizing the difference in those phenotypes based on the genotypes that we get from field caught mosquitoes. As opposed to the Culex mosquitoes, once I do the topical bioassays, it kills a lot. Naturally, it kills a lot of the mosquitoes, and I'm not able to perform these colorimetric enzymatic assays, enzymatic activity assays with dead mosquitoes. I can only use the live controls, so I have a much smaller data set, or a much smaller sample of these, but I'm also able to use mosquitoes from sampled from leftover from my cages. So we're specifically looking at GSTs, cytochrome P450, alpha carboxylesterase, and beta carboxylesterase, which are all known to reduce the amount of insecticide once it's applied as a resistance mechanism. These are just some early preliminary data graphs that I have from sites on which I performed many of the topical bioassays. I haven't gotten to the enzymatic profiling just yet. I'll be working on that more so in the spring. And I guess this isn't too useful just yet, because eventually I would like to overlay these graphs so you can kind of compare which sites are more resistant than the others. But basically, I've been applying certain doses to each of these mosquitoes at certain sites, and I was able to establish what's called an LD50 with just this preliminary data. I have a lot more topical bioassays to perform, and you could see, at least from these graphs, the tick marks that are outside of 20, 50, and 200 are what I placed as the LD50. So this one's like approximately 56, 57. This one's 67. This one's 132. So this, at least this RT235, based on the data that I've collected already, you would call that more resistant than the other sites we have here. And again, this is just preliminary data. Right now, we picked all of our field sites based on Maricopa County abundance and fogging data from the past 5 years. So each of these sites were actually chosen with the qualification that 3 of them would be at a no fogging at all site, about a medium fogging site, and a high fogging site. So eventually we'll be able to hopefully correlate that with the doses as well. But with this data, we'll be able to use this to work with the enzyme activity assays and see if we can establish some sort of relation between the LD50 and what's going on at the molecular level. Moving on to the work that I'll be funded with the PacVec project, my operational field and semi-field studies for insecticide contact efficacy. We really kind of have an incomplete understanding of insecticide contact efficacy in an operational setting as a function of both land cover and as a function of residual spray on varying substrate type. And so what I'm trying to get at is, when vector controls go out and spray either with a plane or with a fogging truck, they spray at a certain dose, but we really don't know how much insecticide is penetrating into the areas where that are not immediately adjacent to the road. So what I really would like to know is how the physical dispersion of insecticide droplets are influenced by both the operational limitations of fogging machines and how the varying environmental substrates influence transmission of insecticides to resting mosquitoes. And that's my first aim. For my second aim, I'd like to see how land cover type and environmental barriers affect airborne contact with Aedes aegypti mosquitoes susceptible to insecticide, and possibly also susceptible Culex mosquitoes. So my methods for the first aim of my project is to set up a semi-field study. Generously, but with Maricopa County, they have a contact with a wide open field in Maricopa County where we can perform, and they perform open insecticide fogging studies. So we'll be able to set up a grid, which looks a little bit like this, with different at different points, at different distances from the road. So at least you can see the driving direction and the spray line going up this vertical arrow, and then the distance from the road as from this horizontal line right here. But just to make our lives a little bit easier, we'll probably limit the amount of sites or amount of points that we have, because we have to generate all of the mosquitoes to populate into these cages. And then after the spraying truck drives by the setup with both these cages and these glass slides, which will be used to capture insecticides, we'll assess the mortality within the cages and quantify the insecticide droplets on the glass slides with a UV dye spectrophotometry. In addition, going back here, within these points, we'll also set up different objects of different substrate types. So maybe some bricks, like pieces of wood or tables, different plants, in order to collect the insecticide that's sprayed onto these different substrate types, and to assess the insecticide resistance phenotypes on the various substrates. We'll use the WHO cone bioassay, which is basically just a plastic cup that's strapped down to whatever substrate that you have. You put about 25 or so mosquitoes inside and let them be exposed for an hour or so, and then assess their mortality at a certain time point. So we're really trying to look at just basic things that would be found around either people's backyards or just in general, like brick walls or stone walls. So at the present time, we're going to look at like wood, leaves of pretty common plants in the Maricopa County area, and then probably some kind of plastic. And then using that data from AIM 1, we will then try to assess the insecticide fogging efficacy over different land cover types using an operational field study. And the way that's going to differ is, at least for the semi-field study, we'll be working directly with Maricopa County. They'll join us in the field, and they'll be able to spray in a targeted way where we tell them to. As opposed to this study, we're going to be doing more opportunistically, where the county will be spraying at a certain time. They'll give us about a 1 to 2 day notice of where they've gotten certain exceedances. An exceedance is just when the amount of mosquitoes has exceeded a certain threshold via one of the 800 plus traps that Adele was talking about, or they had a West Nile virus positive pool in the area. We'll set up our grid with with about 15 points, just like the other in AIM 1, but in this time we'll be more at either non-random sites that are placed, or points that are either placed like behind trees or different land cover barriers like trees or walls, some that are kind of in the open area as kind of a control, and then some in random spots. And then again, we'll also be placing cages with susceptible Aedes aegypti and or Culex quinquefasciatus and glass slides to capture insecticide droplets. And we'll be using this data in order to create a spatial contact efficacy model. Yeah, this is just what I talked about, but a special an efficacy model with some collaborators that we had just found based on this paper that I had read about using Google Street View images to create heat maps for, at least in this paper, they used it to make heat maps based on land surface temperature. But what we're looking at is to see if we can use the street level data that we're collecting from our experimental setup and convert that into a heat map that will allow us to visualize the efficacy of insecticide across a different landscape. So as you can see, just on a basic level, this is actually really complicated computationally, but using Google Street View images, they're able to be converted into these different land cover types and sky. So you could see, as you could see, the gray is building, green is tree, black is impervious. So that would be like walls or anything that's put up. Pervious would be like sand or anything that something is able to be passed through, and also moving objects. And then using the flat images is able to be converted into a sphere and then overlaid onto a map. At least this map right here is showing how the green, the tree fraction, varies over a different land cover area. And we'll be using this in combination with aerial remote sensing imaging, as they do in the paper as well, to create hopefully something that looks like this. This is the land surface temperature map that was created from the paper. But we would like to look at to see where the best place or the most ideal places to spray to get maximum efficacy from a fogging truck. All of this to say that there is very likely different efficacy in urban areas, suburban areas, and a rural area such as this in the Southwest. And we'd really like to know how efficacious our insecticides are, so that we can better quantify how resistance is changing in the area, and also how efficacious our efforts are when we go out and spray mosquito populations and reducing the abundance in the area. So the real purpose of my work is to provide a framework for Maricopa County Vector Control and other mosquito control agencies to better understand and target populations of mosquitoes to reduce resistance allele frequency on 2 spatial levels via their genotype and their enzyme profiles, or by their mortality phenotypes with the dosing assay, and also spatially via the most optimal locations at and in surfaces on which to spray to achieve maximal insecticide contact efficacy. I'd just like to thank PacVec for providing me with the funding to do at least the second half of this work. My lab and Dr. Pyman's lab, the Maricopa County Vector Control District for providing me with 5 years worth of data to run through and helping me with placing my traps, or guiding me with places to place my traps, as well as helping me out with the work that I'll be doing in the upcoming spring. And then our 2 collaborators that we'll be working with to help us out with the spatial modeling. And that's the end of my talk. Does anybody have any questions?
Thanks so much, Don. That's a great talk. Who has questions? I know I do, but I'll let somebody else go first.
Oh, I see there's a lot in the chat. I didn't even notice.
All right. And if you close your screen too, we can see people like raising their hand and stuff.
Let's see.
I know Chris has a question in the chat.
Let's see. Related to just going to say it out loud. Related to Kim's question about oh no, that's for Adele, sorry. Don, really interesting talk. For one of the sites in the dose response curves, the mortality seemed to flatten out at a minimum of around 20%, even at very low doses. Do you know what could be different about that trial or population where you're able to compare the bioassay dose response curves for unknown field samples and with known susceptible colony mosquitoes. At least for the second part of your question, Chris, we have susceptible colony mosquitoes, and that's one of my next steps. Probably in the spring, once we get a little bit of time, we'll also do the bioassays on the susceptible colony mosquitoes we were given from our collaborator in Florida. I have to look at what you're talking about with the curve that you were talking about. Can you tell me which one you were talking about specifically?
Yeah, sorry for doing it in the chat. I was expecting maybe other people
Yeah.
wanted to ask, but yeah, it's on that one slide where you're showing the dose response curves. I think it was the curve on the right middle, maybe.
Oh yeah, yeah.
And I don't know what the site codes mean, but that one seemed to just sort of flatten at 20% and stay at 20% regardless of the low dose. And I was curious if you saw that in your assays where certain populations were just dying at some rate as a baseline.
So I mean, these are controlled. Like there's a correction that we do that factors in the control mortality. So I guess the answer to your question is no. That we do. I mean, there's no other than the control mortality that we have. There's nothing else that goes into this data, and I haven't really seen if there's any other kind of difference there that I
Okay,
can
that's one reason I was curious about the
Yeah.
susceptible colony mosquitoes, where if they're paired, you could
for
see
sure.
that
Yeah,
potentially.
definitely. We'll definitely have to compare that to the susceptible colony mosquitoes, because at least from our work with Aedes, there's a lot of sites that are very, very resistant in this area that we know of already. So I'm sure that's pretty similar for Culex as well.
Right, that's actually what's going to be my question. So we did a study earlier, which we still need to get published. I swear I'm going to do it during my sabbatical. But we did not find any susceptible populations of Aedes aegypti to any of the pyrethroids. We did not look at malathion because we were just looking at what Maricopa was spraying. And even though they were confident that they were still getting some kind of operational effect with deltamethrin, when we did, and we just did the good old fashioned bottle bioassays, but they were all highly resistant. And so that's one thing I was wondering is at this point, is space spraying even going to work for Aedes aegypti in Maricopa? Are there, with the places that we saw, we went where the Aedes aegypti was, and they were highly resistant?
Yeah, at least from the data. So like I was saying, we have another project going on that's doing a much wider surveillance, and also with our collaborator of Aedes aegypti, resistance genotypes in the area. From the little I did and from the data that I've looked at, I agree with you. Like there's a lot of resistance in Aedes aegypti, and it probably isn't very effective, the spraying that we do. But I have 4 out of the 6 sites that I was sampling last year were intentionally sampled to try to find genotypes that were not as resistant, and so they're out there. There are small susceptible populations out there that are only slightly diluting the resistant populations. But yes, in the county, well, I think I would say a lot of the prevalence is definitely resistance.
Yeah, I mean, we were to the point of wondering whether it's basically a metapopulation that already had resistance genes at a high frequency before it even reestablished in Phoenix.
Right.
Whereas when we looked at quinks, we didn't look at tarsalis, but when we looked at quinks in the same area of the southeastern part of Maricopa County, there was a lot of resistance to pyrethroids too, but it was not as pronounced. And you could certainly understand why they were still getting some degree of operational control. But with Aedes aegypti, we did this whole study looking at population density before and after spraying and age structure before and after spraying, and there was no difference. And the smoking gun for us was just a high level of resistance. But that was in the southeast part of the county, which, as you said, is sort of a hot spot.
Yeah, at least for the doses that I was working with, the doses that I'm working with for Culex compared to Aedes are like 20 times higher. So the Culex are, like, at least for deltamethrin, it's a lot more resistant comparatively. They require a higher dose to actually kill that 50% of the population. So,
Right.
yeah.
Well, I really want to talk to you more about this, but I know that we have reached our 2:00 time. Maybe do we have time for one more question? Jennifer, did you want to say your question out loud or just in the chat?
Oh, it's not a question, so Kathleen, you should go ahead and
Oh.
ask it.
Thanks.
Anyway, maybe I guess if people have other questions for our speakers, thank you both. Those were both really, it's exciting to hear this work. I don't know what the situation is with the recording. Maybe Chris, you can tell people how they can access that, or other people can access that.
Yeah, it will be on the PacVec website, and Celia is posting these as we go. And then also when the next seminar comes around, if you're receiving those notifications, she includes the links in the email. All the magic of Celia, of course, but thanks for hosting, Kathleen.
Well, thanks, Don, and thanks, Adele. I learned a lot.
Thank you all.
Really
Appreciate it.